Good morning, everyone. My name is Sebastian van Nuffel. I'm an assistant research professor at Pennsylvania State University. And first, I would like to thank the people from Physical Electronics for inviting me to give a talk during their virtual fall workshop. The topic of my talk will be our newly published paper in the Journal of the American Society for Mass Spectrometry, titled Chemical Imaging of Retinal Pigment Epithelium in Frozen Sections of Zebrafish Larvae, using time-of-flight secondary ion mass spectrometry. Let me first introduce my collaborators, my co-authors, Professor Qi Chang, Dr. Kai Ang, and Dr. Alex Lin from the College of Medicine at Penn State. Qi and his group conduct research on zebrafish, which is a very important model in biology and in biomedicine, because the zebrafish shares a lot of genetic material with humans. And over here you can see the developments of the zebrafish from larva, from embryo to larva to adult. Qi and his group mainly investigate the morphology of these model organisms using X-ray micro-CT, and this technique allows them to visualize in 3D the entire organism. Now, a couple of years ago, in 2005, Qi published a very significant paper where they compared the genome of the wild-type zebrafish with that of the golden mutant. The golden mutant is a lot paler compared to the wild-type. And it discovered that the golden mutant had a genetic mutation in the SLC24A5 gene, which they presume encodes a potassium-mediated calcium exchanger. And the funny thing is that that same gene is also present in humans, and Caucasians also carry a mutation in that gene. So the funny thing is that a genetic mutation in the same gene has the same effect on both humans and zebrafish. They become more pale, they become less pigmented. Now, what you really need to do to prove that you're knocking out this calcium exchanger is to have a look at the calcium concentration in tissue that is rich in melanosomes, for example, the retinal pigment epithelium in the eye. X-ray micro-CT, however, is not capable of chemical imaging. It's very good at three-dimensional volumetric imaging, but not chemical imaging. And that's where our collaboration started, because TOF-SIMS, time-of-flight secondary ion mass spectrometry, is capable of label-free chemical imaging with the same kind of spatial resolution as an optical microscope. It's capable of imaging elements, inorganic materials, organic compounds, and a lot of different biomolecules. It's especially good for relatively small molecules, such as lipids and metabolites. It's also capable of looking at drugs and toxins. It's not good for large biopolymers, such as proteins and DNA. They tend to fragment into their building blocks. But the first step, of course, is to figure out how exactly do we get retinal pigment epithelium of a zebrafish larva in an ultra-high vacuum environment. And then once it's there, what sort of molecular markers can we use to actually identify which cells are retinal pigment epithelium? And that was the subject of the paper we just published. We started off with our zebrafish larva. We expect that the effects will be largest in larvae compared to adult zebrafish. So we start off with larvae. They're about 2 millimeters in size. And we line them up in aggro swells, which we then filled up with a 20% BSA matrix. And they are all aligned the same way, so that during the sectioning with the cryo-microtome, you should be able to get a good cross-section of one of the eyes. These thin tissue sections were then mounted on top of indium tin oxide-coated glass substrates, which we had to cut to 90 millimeter discs to fit the phi cryo stage. And it is important that our glass is indium tin oxide-coated, because we need to have a conductive surface for an efficient ion extraction. We then conducted some cryo-TOF-SIMS, so we kept our samples frozen, so they were in a frozen hydrated state, which is as close as possible we can get to the native states of the tissue. We used a nitrogen-filled glove box to prevent the absorption of moisture from the air, because otherwise you would form a thick ice layer on top of our sample. We're not able to do our surface imaging. And at the same time, we cool down the stage down to a temperature of about -145 degrees centigrade. This will allow the tissue to remain in a frozen hydrated state without any sublimation occurring. And it is important that we measure our samples in a frozen state, because when samples freeze dry, small molecules and ions tend to diffuse. And our eventual goal is to image calcium in the retinal pigment epithelium, and we don't want that to move around. After our experiments, we freeze dry the tissue in its optical microscopy. And when we have a look at the optical microscopy image, we can see that our sectioning was quite successful. Over here we have the indium tin oxide coated substrates. This is the BSA matrix. This over here is the zebrafish tissue. This is the eye lens, and then this black boundary layer is the retinal pigment epithelium. We took some secondary ion images. So in TOF-SIMS, every single pixel represents a full mass spectrum. And we measured both positive polarity and negative polarity ion images. So we have two very large data sets, and normally these are analyzed separately. But I thought to myself, this is spatial information from the same region, so we should be able to turn this into a single hyperspectral data set, and then perform some kind of statistical analysis on the entire collection of mass peaks we detected. That's exactly what we did. So we took positive and negative polarity ion images, digitally aligned them, and create a single hyperspectral data set for principal component analysis. Principal component analysis is an analysis method that looks at correlations between mass peaks, and then calculates a score that tells you how many of those mass peaks are present in that particular region. So in this case, we can see that the first principal component separates out this area from this area. And the red area, which corresponds to the negative loadings, is all the frozen material we're looking at. The green area clearly colocalizes with the indium tin oxide substrates, and therefore the positive loadings contain a lot of indium tin oxide peaks, and we can see these repeating clusters of indium oxide. The second principal component seems to separate out something else. Looks like it separates out the zebrafish tissue from the BSA matrix. And as we can see in the positive loadings, we have a lot of phospholipid markers. Phosphocholine head group, phosphate in the negative polarity. But that all makes sense. And then the negative loadings contain a lot of water cluster ions. So we can see repeating clusters negative of ice, which corresponds to the red area. And when we create a couple of ion images for some marker ions, we can see that the indium tin oxide image is a negative of the ice image. And the phospholipid image partially overlaps with the ice image. And this is good news because this clearly demonstrates that our tissue is still in a frozen hydrated state. So this means that the experiment is successful. We have successfully sectioned through a zebrafish eye, kept it frozen, put it in our instruments, and the material is all still there. Then when we delve a little bit deeper into our data, for example, principal component number 7, we see that the positive loadings correspond to an amino acid signal. So we can, in the negative polarity, we can see CN- and CNO- correspond to the peptide backbone. And we can also find residues for individual amino acids. The interesting thing is when we plot one of those amino acids marker ions, we can see that the amino acid signal comes from the tissue, which makes sense because tissue contains protein, and also from the BSA matrix, which makes sense because BSA is a protein. However, there appears to be an accumulation which co-localizes with the eye lens, which at first seemed a little bit strange, but then we realized that vertebrate eye lenses are composed of fiber cells, and they form these concentric layers. And these fiber cells produce very large amounts of crystalline protein. And they produce those types of protein because the islands need to have a high refractive index. And you'll mainly have aromatic and sulfurous amino acid residues. And then when we look at our TOF-SIMS data or chemical imaging, we indeed see an accumulation of methionine, tyrosine, all the aromatic amino acid residues, phenylalanine. So this all still makes sense. Then when we delve a little bit deeper into the data, we have our principal component number 13. And now we're starting to see something that we've been looking for. We've been looking for markers for the retinal pigment epithelium. And this co-localizes with retinal pigment epithelium. So what kind of ions do we find in our positive loadings? Well, we find known melanin markers from the literature. So these are a couple of low mass markers, C3N-, C3NO-, C5N-. And these were already known. However, we also found very strong correlations for positive ions at 152, 174, 190, 303, and also a negative ion at 150. And these have not been reported in the literature. So I got us thinking, where exactly do these ions come from? Because it has to be something that you can only find in the retinal pigment epithelium. And I got us thinking, it probably has something to do with melanin. So we had a look at the biosynthetic pathway for eumelanin. And out of all these different molecules, this guy stood out, leucodopachrome. Because if you take leucodopachrome and lose the CO2 through neutral loss, you get a molecule that has a mass of 151. And in that case, all of those mass peaks we detected and could not explain can be explained as adducts of this particular precursor. So we have the protonated ion, sodiated, potassiated, dimer with a proton, and proton subtracted negative ion. So all this fits together very nicely, so we can be quite certain that this is probably leucodopachrome that we're looking at. And then when we compare our known melanin markers, which are low mass fragments, we see that there's a very high background, and that those ions also appear elsewhere in our image. And that makes sense because these are low mass fragments, which are not very specific. They can be produced by a wide range of different molecules. Then when we have a look at our presumed pseudomolecular ions of leucodopachrome, we can see that it uniquely localizes, or almost uniquely localizes, in the retinal pigment epithelium. So we are able to visualize the retinal pigment epithelium with a mass peak that we can attribute to a molecule that's part of the biosynthesis of eumelanin, an intermediate in the biosynthesis of eumelanin with much greater specificity. And this is exactly what we're looking for, a way to visualize the retinal pigment epithelium in zebrafish larvae. So we more or less succeeded in what we set out to do. Here's a little overlay of all the different chemistries we detected, so you can clearly see ions that we attribute to the ITO, ions that we attribute to phosphocholeins correlating with the zebrafish tissue, ions present everywhere, also come from the BSA, protein coming from everywhere, accumulating in the islands, and then our blue circle, which is the retinal pigment epithelium. So by simply looking at the chemical information, the chemical imaging information, we're able to recreate an image that looks very much like our optical microscopy image. And now for the conclusion. So we successfully established a protocol for cryo-TOF-SIMS measurements of frozen hydrated zebrafish eye cross sections. There's also a protocol that we can expand to other types of tissue. We also demonstrated through advanced data analysis and by combining our data sets into a single hyperspectral data set and then combining that with multivariate analysis, we can really enhance the data interpretation. We're also able to visualize the islands because of the crystalline proteins. We were able to visualize the RPE using pseudomolecular ions indicative of leucodopachrome, intermediate in the biosynthesis of melanin. And now we have all the tools in our hands to investigate the function of this SLC24A5 gene and start comparing the calcium uptake between their epithelium, between wild type and mutant zebrafish larvae. And this is where I will end my presentation. I'm very happy to answer any questions people might have. Thank you very much.